Greetings everyone, we are from Group 6, Subgroup 1, and this is our video that is Lab 1 Spectrophotometer Protein Analysis. Hello, I am Nituli Denisiawo and my number metric is 105341.
Hi, my name is Ilararo Panadoni and my matrix number is 107686. Bye.
Hello, my name is Vina S.C. from Anak Brian and my matric number is 104191.
Hello, my name is Nur Afika Alia binti Awang and my matric number is 107696.
Hello everyone, my name is Eviran Tasha Alamongi. My matric number is 107529.
Our objectives are to learn how to use a spectrophotometer to identify biological molecules in qualitative analysis and determine their concentration in quantitative analysis in a sample. And our materials are spectrophotometer, cuvette, Bradford reagent, distilled water, test tubes, rack micropipetters, BSA protein, and standard unknown protein solution A, B, and C. Our experimental procedure is: before conducting the experiment, we were asked to wear gloves for safety purposes. Next, we place 4 ml of distilled water in 7 test tubes. By that, we use micropipetters to put it in. Then, we place 1 ml of Bradford reagent into each tube except for the 7th tube. After that, 50 microliters, 100 microliters, and 200 microliters of BSA protein was added and has been prepared in duplicate. Then, after that, each tube is shaken using vortex test tube mixer, so that the samples can be read accurately using the spectrophotometer. Before adding into the spectrophotometer, we transfer the liquid into a smaller place called cuvette. In this part 2, the procedure is the same as the part 1, but only the difference is, instead of the BSA protein was added, the unknown protein concentration of A, B, and C absorbance was added instead. So each of the concentration, for example, concentration A has 2 tubes, concentration B has 2 tubes, and concentration C has 2 tubes. After that, the same as the part 1, each tube is shaken using vortex test tuber mixer. So, the samples can be read accurately using the spectrophotometer again.
To conclude, we can successfully demonstrate spectrophotometry and Bradford method for measuring protein concentration, then use standard BSA solution to create a standard curve and calculate unknown samples. This is to proven effective for both qualitative and quantitative protein analysis and gain hands-on experience with micropipette sample prep and spectrophotometer use. Lastly, we can achieve the objective of applying spectrophotometry in biomolecular analysis. There is some challenges that we were facing while doing the experiment, which is the pipetting accuracy, which is the errors in pipetting affected concentration consistency and absorbance values. And next is the inconsistent mixing, which is the poor mixing of BSA with Bradford reagent led to absorbance variability. And lastly is the air bubbles that trap in cuvettes of wells that cause inaccurate absorbance readings. For our upcoming experiment is to request for clearer guidance on Excel by step-by-step instruction for graphing, statistical calculations, and data organization. Next is to improve understanding of experimental materials by in-depth explanation of properties, applications, and handling techniques that include chemical composition, practical limitations, and best practices for precision and reliability. There is some new learnings that I gained from the laboratory session. First is to learn how to use the laboratory equipment properly and safely, such as the automatic pipettes, micropipettes, and spectrophotometers. Next is to gain skills in solution preparation techniques, extracting biological materials, and proper use of control samples. Third is to develop practical data analysis skills on interpreting experimental results and comparing results with reference values, theoretical concepts.